So tonight we have a special treat. As you can see, we have a lab set up here. And we invite you to our classroom where two of our scientists will help explain more of these breakthroughs taking place beyond my lab at the Providence Cancer Institute. So please join me in welcoming Dr. Bernie Fox and Kristina Young, along with Laurel Porter.
Thank you, Dr. Weinberg, and welcome to Dr. Fox and Dr. Young. Welcome to all of you to our classroom, Immunotherapy 101, where we'll have a better chance to understand some of the research happening every day at Providence. But first, let's get to know our professors a little bit. Dr. Fox, you've been at Providence for 26 years. Why did you choose Providence?
I'm here because of Walter J. Erba, Dr. Erba, who's sitting out there. Yeah. 26 years ago, next month, I met with Dr. Erba his last weekend on call at the National Cancer Institute. That was before he came to Portland. And he shared with me this vision to develop in Portland, Oregon, a cancer immunotherapy institute that would do cutting-edge, early-stage clinical trials in patients. And he suckered me in to come to Portland. And today, that vision, supported by our donors and the incredibly hard work of the scientists and physicians, the nurses, the staff, have let us develop an institute that is internationally recognized as a center of excellence. And people use those words, I think, a lot without knowing what they mean. But when I say that, what I mean is that our center has more than 100 clinical trials that are open for almost any type of cancer that you might have. And that the investigators that are there are internationally recognized and they're sought after to speak around the globe. I will point out that one of my colleagues who's in the audience, Dr. Rob Widener, came back from Portugal on Monday. Dr. Brian Bell, who was here last year, is in Rio de Janeiro to give a special presidential lecture tomorrow in Brazil. And I'll get on a plane tomorrow to go to the Royal Academy in London, in England, to talk about the work that we do in little old Portland, Oregon. So it's that kind of level of the clinical trials and the people that we've got here, including that schmuck Andy Weinberg, that wonderful guy, Dr. Andy Weinberg, that bring the bright young investigators like the beautiful Dr. Kristina Young, who's here, who are really focused on doing exciting new phase 1 clinical trials, early phase clinical trials, and taking their science and moving it from the bench to the bedside. So that's why I'm here. I'm here because of all that.
We're so glad that you're here. Let's talk
here.
about Dr. Young. Let's hear for Dr. Fox. Dr. Young, why did you choose Providence?
I would say that Dr. Erba is a key factor in me choosing Providence as well, and I think that his vision of creating this institute that rapidly translates from the bench to the bedside was really a draw for me. I've always wanted to see patients and treat them in our radiation oncology department, but to also be able to run a lab and develop new treatments that I can then take to my patients. And this institute was one of the best places in the world to be able to do that. And I'm just so grateful that we're able to have trials open here that are available nowhere else in the world. And to be able to have a hand in developing those new treatments and technologies that will change the standard of care for patients, both in Portland and across the globe.
Well, that is inspiring. We're lucky to have you both here. Thank you. And I think we established Dr. Erba as a rock star. Attracted these guys here. Thank you for being our professors tonight for Immunotherapy 101. We're going to have the chance to better understand immunotherapy, the main focus of Providence cancer research efforts, and teach everyone a little bit more about the current research going on right now, every day in Portland. So let's jump right in, Dr. Fox. We know that immunotherapy is the process of activating the body's own immune system to fight cancer. But what's really going on here, Dr. Fox? Can you give us a better understanding of how cancer hides or turns off the immune system and the work you're doing to try to counteract its efforts?
Sure. So, thank you, Laurel. So, unless you've been under a rock, I don't see how I can get to this. Okay, there we go. So this is from the Wall Street Journal, but you've seen it in every magazine, on every television channel. There have been advertisements about these sorts of stories, about patients with cancer who have failed all standard traditional therapies that have been called super responders. And it's this, how the promise of immunotherapy is transforming oncology. And this is the vision that Dr. Erba had 26 years ago or longer than that many of us had that have been in this field for a long time, that this could have a huge impact on patients' lives. And it has made a huge difference. And in fact, patients who have achieved a complete response with immunotherapy, and specifically with IL-2, which is a drug that Dr. Brendan Curdy still uses at the Child's Research Institute at Providence, patients who achieve a complete response with that drug have more than a 20-year survival rate without any cancer. So that means you're curing patients of that disease. We don't know if these new therapies are going to do the same, but we're hopeful that they will. So what I'd like to just point out that in the last in December of last year, the Nobel Prize in Medicine and Physiology was awarded to 2 investigators for the discovery of checkpoint blockers. That is the drug, those are the drugs that have led to this revolution in how doctors treat a large number of cancers today. So if I go to this point, right, go to the next point, I want to take you through the story of how cancer evolves to escape the immune system. So many cancers, melanoma is a great classic example of this, but many cancers can shut off the immune system so it can't recognize it and destroy it. I'll just point out that we think everybody develops cancer over their lifetime, but those of us that have got their immune systems are revved up or our tumors are not as smart, those tumors get killed. But if the tumors are smart and can become stealthy, they can evolve to escape us, escape our immune systems. So how does that work? So here's a cancer cell in an immune cell, or a cancer killer cell. What many cancer cells can do is they can express a molecule called programmed death ligand 1, or PDL1, on their surface. What that molecule can do is it can bind a cancer killer cell and turn it off. When those two things bind together, that T cell can no longer kill the cancer cell. So what happens is that the cancer cells now grow and multiply and divide. They escape the immune detection. So the drug for which the Nobel Prize was awarded, what it does is that drug is it's a protein. You get it by an IV infusion at a clinic. And what that does is it goes and it binds to this molecule called PD-1 on the cancer killer. Can you go back? Can you go back 2 slides? I got it right here. I'm touching the slide when I'm not supposed to. Okay, so when that binds the cancer killer cell's receptor, this can no longer bind and turn off this T cell. So instead of letting the cancer cells grow, now the cancer killer cell can go in and kill the cancer. That's the revolution. That's what's changed, fundamentally changed, how we treat a large number of cancers today. And so you look at it, whether it be lung cancer, head and neck cancer, non-Hodgkin's lymphoma, breast cancer was just approved. This is what the main treatment modality. And it's not perfect, but in many cases it doubles the response rate over standard chemotherapy. So, what does it look like? So here's a patient's cancer. So these orange cells over here on the right are the cancer cells. I'm a little afraid to touch the screen again. So those are the cancer cells. The cancer killer cells are the guys in green now. So you can see they're separated by that wall of red. That red is that PDL1. That's the molecule that can shut off the cancer killer cells. So this patient, if you looked at this patient, had a biopsy before they got treatment, you'd say this patient, this is the patient that should respond. If you can block that interference, these immune cancer cells can get in and kill the cancer.
We got a little sneak preview, Dr.
Yeah,
Fox.
I know.
Show us what it looks like in a patient less likely to respond well to immunotherapy.
So while this patient is expected to respond because they got the cancer killers here, they got the PDL1 molecule here, these guys over here don't have those immune cells there. They've got very little PDL1 on the surface, and there's only a few of the cancer killer cells that are present. So this patient, based on statistics, is less likely to respond to this drug. They need something else to boost that immune response. So I'm from Detroit. The automotive used to be the automotive capital. Pistons didn't do so well. I was glad that we beat them here. But so the idea is that the biggest blockbuster drug in oncology is PD-1. PDL1, that blockade. That's thought of as a brake because it's removing the brake. Remember that PDL1, the cancer cell expresses PDL1, it binds the cancer killer cell, it shuts it off. That's a braking system. So the first drug that you talk about, actually, Opdivo, Keytruda, Yervoy, those are all things you see on television. Those drugs take the brakes off the immune system so it can kill the cancer. There's another wave, and Dr. Weinberg played a critical role in discovering this, and Dr. Brendan Curdy and Dr. Erba worked on the clinical trial to put it first into humans. That was a family of drugs called agonists, or T-cell agonists. They're gas pedal drugs. What they do is when the car is turned on and running, they give it the gas so it can go faster, so it can run into the cancer, take the brakes off, it can kill the cancer. That's the concept behind it. These two drugs are currently in clinical trials. People are combining these two drugs together. And in some patients, they have a better effect. But it's still pretty low. The response rates are not as what people would like to see. So still, the vast majority of patients still fail to respond to immunotherapy. So what we think, based on a lot of animal data for a number of years, is that in many cases, the car is not turned on. There aren't many T cells or immune cells that can see the cancer. So there are many strategies to talk about, from cancer vaccines. You're going to hear from Dr. Young in a little bit about another inhibitor that she's been studying that inhibits this. But things like chemotherapy and radiation that are common therapies for cancer, they can have synergistic effects or may have synergistic effects, so they can be combined with things like OX40 and then with anti-PD-1.
Well, we know that Providence, Doctor, is known for trials worldwide. Can you tell us about a couple of clinical trials happening right now that our students might be especially interested in?
So we're very excited about this next set of trials. So we have two trials that'll be the first on the planet to do this, which will combine a complex vaccine, which was actually developed out of Providence Technology. Dr. Hongming Hu developed this technology. It was patented, and one of the spin-out companies that Laurel had mentioned, We'll Be Back, has got the vaccine. The gas pedal, which is OX40, which Dr. Weinberg talked about briefly, or was talked about there, which Agonox license and AstraZeneca was developing, and then giving it a break. So we're going to give all three, and it'll be the first time that they've given all three, something that turns on the car, gives the gas, and hits the brakes. Those studies will start in advanced triple-negative breast, for which advanced triple-negative breast cancer is today a death sentence. There's not a curative treatment for that disease. And we think, based on lots of preclinical data and early data, in fact, the FDA approved anti-PD-L1 for women with advanced triple-negative breast. But as Dr. David Page, who's in the audience, will tell you, the patients are still progressing after time. So that trial that Dr. Page will lead will hopefully open at 6 different centers in the United States, which will launch with a partner, which will launch later this summer or early fall, we hope. A second study, which will be an advanced head and neck cancer, will be led by Dr. Ram Leidner and Dr. Marcus Cooey, who are here. But we'll take, the vaccine with a gas pedal drug and a checkpoint blocker to put all three together. And it may be hard to believe, but this is happening in Portland, Oregon, and it's not happened yet anywhere else on the planet. So it's kind of unbelievable, and it's because of your support that it's here.
You know, listening to you, it sounds like the future really is personalized medicine.
I think if you look at you saw posters here tonight at several centers, you saw videos of T cells killing cancer. There's much going on in the field, but it's our ability now to take the molecular work that Dr. Balfuco does and some of the things Brian Pining has been talking about and what Eric Tran has been doing, putting them all together to come up with a tailored therapy, very precision immunotherapy. This patient has got T cells, all they need is PD-1. This patient doesn't have it, they need something else. Dr. Young's going to tell you about what she does and her molecules and things she can identify that's going to give us other treatment methodologies for patients. So it's an incredibly exciting time, but I'll stop there. So
Thank you, Dr. Fox. Dr. Young.
Thanks. There you go, Dr. Young.
So we're very excited that we have a trial open here that will be addressing some of the things that Dr. Fox just talked about. So the trial that we have open that's available only here at Providence is targeting one of those braking mechanisms, and it also gives the immune system a little bit of gas too. It's a molecule called TGF-beta, and we're combining a TGF-beta inhibitor with chemotherapy and radiation in patients with rectal cancer. And the goal of this trial is really to turn some of those cold tumors where there's a poor immune infiltrate into hot tumors and really get that immune system involved in taking care of that patient's cancer before they go to surgery. So what you see up here is evidence of a cold tumor. So this is cancer cells in green and the T cells that kill the cancer cells are in yellow. And this is a patient's biopsy before they enrolled on study. And you can see that there's very few immune cells in this patient's tumor. After getting 2 weeks of the TGF-beta inhibitor, you can see that we're starting to get a few more immune cells getting into the tumor, but it's still not what we're hoping to see. But what you can see, by the time this patient goes to surgery, this is truly now a hot tumor. Those green tumor cells are being swarmed by the anti-tumor T cells, and this patient is having an outstanding response to therapy for a very advanced tumor that had a poor prognosis. So what we're seeing is we want the radiation and the chemotherapy to start the car. And then we're using this TGF-beta inhibitor to give a little bit of gas and take the foot off the brake so that we get a much better response.
So those 3 images that you showed us, are those 3 different biopsies on the same patient?
Yeah, and that can be really hard on patients already going through cancer treatment. So right now, one of the state-of-the-art technologies that we're using is this multispectral imaging that gives us these beautiful pictures and insight into what's going on in the patient's tumor. But that's also really invasive. You're asking a patient to go through 3 procedures to get these biopsies. So one thing that we're working on is utilizing MRI technology as a non-invasive measure of what's going on in the tumor microenvironment. So what we're measuring with the MRI is not the same exact thing that we're seeing under the microscope, but we are seeing that these 2 things correlate. So when we have an MRI image that looks like this, the tumor outlined in the white dotted line, this is a rectal cancer, we see that this is likely a patient that has a low immune cell infiltrate, so a cold tumor, unlikely to respond to standard therapies. When we see an MRI that looks like this, we think that this correlates with a hot tumor with lots of anti-tumor T cells, much more likely to respond to therapy. So now we think we know if we were to biopsy these patients, what we would see under the microscope. And so that's giving us the ability to evaluate what therapies might be best for the patient without having to poke them with a needle, which is what the patients really want. So thanks to the generous donors, we now have this new technology that's being installed in our radiation oncology department right now. This is an MRI fused with a linear accelerator called the MR-Linac. This should be up and running in our department this fall. We're really excited because not only does this technology allow us to target tumors better, avoid normal tissue better, but it will also provide us the ability to evaluate in real time changes that are happening in the tumor microenvironment so we can really see, are we starting the car? Are those T cells getting into the tumor while we're radiating the patient? So it's something that's unprecedented. It's going to be available here first on the West Coast. We're really excited about this technology for all of the benefits it'll bring our patients. These really are very exciting times. Class dismissed.
Class dismissed.